two major categories we've talked about so far are the antibiotics that inhibit cell wall synthesis, they interfere with peptidoglycan synthesis, or in the case of the isoniazid, the mycolic acid synthesis associated with treatment of TB. And then the other one is protein synthesis inhibiting antibiotics, and we mentioned that typically the two targets for those particular antibiotics are either the 50S subunit or the 30S subunit. So interfering with, on the 30S subunit, for example, with the binding of a tRNA to the A site on the 30S subunit, or interfering with one of the proteins that is responsible for peptide bond formation on the 50S site, or interfering with translocation on the ribosome. That's primarily associated with the 50S site as well. There are some other antibiotics that exploit kind of a third vulnerability of bacteria. And these are antibiotics that interfere with either DNA replication or transcription of DNA into RNA. And both of those are good strategies, because if you can't transcribe DNA into RNA, then you don't have any messenger RNAs to make protein. Interfering with DNA replication itself is a very effective means of killing microorganisms because the DNA has got to duplicate prior to binary fission or the cell dies. There are a couple of these particular types of drugs that I want to mention briefly. The first one has become kind of a relic almost. This is a drug called actinomycin D. And this particular drug selectively binds to DNA, and then it prevents, as a result of this binding, the transcription of DNA into RNA. So this is a transcription blocker. When this drug was first discovered, they tried to use it in some medical treatment, but it's an extremely toxic drug, and the side effects far outweighed the benefits of this particular drug. So it really never made it past clinical trials to really get into widespread human use. But it still has a role. This particular drug played a real role in molecular biology research, because early on when we were figuring out the central dogma and how all of this flow of genetic information worked, it was very interesting to apply this drug to different cells under different conditions and see what effect blocking transcription actually had on those particular cells. So this is a drug that has been used a lot in research, and you'll see reference to it still in some of the more classic molecular biology research articles. An antibiotic that is still being used a fair amount is rifampicin. And this particular drug selectively blocks bacterial RNA polymerase. So it's still interfering with this flow of information from DNA to RNA, but it just does so by a different mechanism. Okay? Bacterial RNA polymerase has some significant differences from the eukaryotic RNA polymerases. There are multiple forms of eukaryotic RNA polymerase, polymerase 1, polymerase 2, polymerase 3. Bacterial RNA polymerase is really different. In particular, it's got a subunit associated with it called a sigma factor that eukaryotic polymerases do not have. And so there's a real opportunity here to selectively target that particular enzyme, leaving the eukaryotic polymerases alone. Another type of drug that interferes with DNA replication is a class of drugs called fluoroquinolones. And the fluoroquinolone that you are probably most familiar with is the drug Cipro. And some of you might have even taken Cipro. Cipro has been prescribed not uncommonly for traveler's diarrhea. Following 2001, some of you may not know or remember that there was a bioterror attack on Congress, and anthrax spores were sent to members of Congress in envelopes, and some of those were released. And Cipro is the drug of choice for anthrax. And there was some concern about whether there was enough of a nationwide supply to provide all the Cipro that might be needed if there was a widespread bioterror attack on the general population. And there were some real interesting questions about who should have access to this. And of course, Congress said, well, we should be number one. There was concern about whether soldiers, for example, would have enough Cipro available if there were bioterror agents released in the Middle East. Fluoroquinolones are kind of interesting. They actually were developed as a byproduct of another antimicrobial drug called chloroquine. Have you ever heard of chloroquine before? Primaquine. Chloroquine and primaquine are anti-malarial drugs. If you travel abroad when in areas where malaria is still a real threat, then your doctor very likely will put you on prophylactic or preventative chloroquine. Did you have to do anything when you went to Africa?
Yeah.
Did you have to take chloroquine?
I think that's
Okay. Well, when they were developing this particular drug, chloroquine, there was some interesting byproducts that were produced. And one of the byproducts of chloroquine production was this compound called nalidixic acid. And as it turns out, this particular byproduct of chloroquine production inhibited a bacterial enzyme that goes by the name gyrase, or is also called topoisomerase. Anybody know what gyrase or topoisomerase do? Back to cell biology. What happens to your DNA double helix as the unwindase enzyme, or the helicase enzyme, starts unwinding the double helix? What happens to the end where the hydrogen bonds are still intact? It's all supercoiled, right? It gets all twisted up and stuff. And these enzymes, gyrase or topoisomerase, make little nicks in that supercoiled region and allow it to basically uncoil so it can continue through the replicative process. And this particular byproduct interfered with these gyrase/topoisomerase enzymes. Well, they thought that was interesting and thought it had some possibilities. This particular compound right here wasn't suitable for use in humans, but they modified this particular compound. And organic chemists added some fluorine to it and some amino groups to it, and were able to develop this new class of drugs, the fluoroquinolones, which still inhibited these gyrase enzymes, but were safe to take. Okay, and so this is kind of the origin of Cipro. So sometimes drugs are developed in kind of accidental ways. And I'm not sure what caused somebody to say, gee, let's test one of these particular byproducts of chloroquine production and see if it might have some antibacterial effects. And then when it did, they had to figure out how it worked. What was the target? And then modifying that biochemically to then make a derivative that was safe for human consumption. It's kind of an interesting little side story. Well, we're going to talk about the development of antibiotic resistance and what a problem it is. But I think you all have heard enough about that to know it is a problem. Drug companies are faced with constantly trying to find new drugs that will treat newly emerging threats and also organisms that have become resistant to traditional methods of treatment, like penicillins. So I'd like to give you a little bit of insight into what new strategies we're trying to employ to develop new classes of drugs that are looking at exploiting different mechanisms than just cell wall biosynthesis or protein synthesis inhibition on 50S and 30S ribosomal subunits, or even the DNA replication. What are some of the kind of things that are coming out on the frontier? It's really important that we get out ahead of this, because drug development, I mentioned before, is a long and costly process. Anywhere from 12 to 24 years, from discovery of a potential molecule until you can go through all the clinical trials which result in the drug being able to be used for human consumption. Billions and billions of dollars. How many times do you hear, well, Lilly stock took another hit because they had this promising new drug in the pipeline, and then it got into phase 3, and the results didn't show any real efficacy of the drug over the placebo, and that's just a killer. And then there are, by federal law, limited patent times. And so once your patent clock starts ticking, you've only got 7 to 10 years before the generic can come in and undersell you and produce the drug for a fraction of what you had to charge. Yeah, drugs are overcharged in some respects, but the general public really doesn't understand the economics of drug development and how much it takes to do the research to get these things through the various clinical trials. So here are some of the things that kind of are on the horizon, or some of these have already been adapted. This is a slide that I actually threw into the PowerPoint, and so if you have an older PowerPoint, you might want to pull these slides out. Or these are in the lecture notes as well. I just pulled these directly from the lecture notes, but I didn't want you to have to sit here and kind of write all this stuff down. Just take a look at what some of the possibilities are. So there's a new drug called daptomycin. And daptomycin is trying to exploit multiple aspects of cell membrane function. Cell membranes are a tricky target for selective toxicity because there aren't a lot of fundamental differences in cell membrane structure of either fungi, bacteria, or eukaryotic cells. Now there are some minor differences, and they're really looking for ways to exploit those particular minor differences. These are an interesting class of new drugs, the oxazolidone class. These are entirely synthetic molecules. Okay? These are built from scratch in the lab. As opposed to the semi-synthetic drugs, like the penicillin derivatives, which use that basic penicillin structure, and then we start tinkering around biochemically with it and modify some of those R groups, for example. This inhibits initiation of protein synthesis. This is kind of a new strategy. What this really does is this blocks the mRNA from binding to the ribosome in the first place. Most of the drugs that interfere with protein synthesis typically interfere with elongation of the polypeptide chain or improper reading of the codons. We talked about some antibodies that fell into this category. But here's a new strategy. Okay? There is one drug, generic name, or named by Zyvox, that falls into this category. And this is really important because this particular drug can be used to treat MRSA and VRE. VRE stands for vancomycin-resistant enterococcus. Remember, vancomycin was always kind of the backup to methicillin. And that was the drug that was kind of held in reserve for methicillin-resistant strains of bacteria. But now we're seeing development of vancomycin resistance. So what's the next drug we're kind of holding back as a last resort? This is one of the drugs. There's some hope that because this drug isn't found in nature, the bacteria will develop resistance more slowly to it than naturally occurring drugs. But that's to be seen. Very important. Physicians are really urged to use this when only absolutely necessary. Okay? Life-threatening situations where you absolutely know that the drug is penicillin, or the bacteria is penicillin resistant and vancomycin resistant. Because the more we use this, the more that we're going to develop resistance. Mupirocin is another drug that inhibits a little bit different portion of the central dogma, and these inhibit bacterial transfer RNAs. So they interfere with actually, for example, the charging of the amino acid to the bacterial tRNA. Same result, you don't make proteins, but a little bit different strategy. For right now, these are only used in topical creams for skin infections, so there's some evidently kind of toxicity issues with internal use for these. Here is a real promising one. Drugs that target bacterial fatty acid synthesis. Metabolically, the pathway and the enzymes that bacteria use to synthesize fatty acid have very fundamental differences from eukaryotic fatty acid biosynthesis. And if we can find drugs that will inhibit those, then that is going to be really big. There is one new drug called plantansomycin, and this is one of the only 3 novel antibiotics that's been produced in the last decade. All the other drugs have been just kind of tweaking of other drugs. Okay? But this is a novel drug that interferes with fatty acid biosynthesis pathways. Another strategy is the use of antisense RNA. In antisense RNA, you develop RNA molecules which are complementary to messenger RNAs. And if you provide those to the cells, they can bind to the messenger RNA, and then that prevents transcription of those messages into protein. I think I did it when you guys were freshmen. Do you remember the video we saw on RNAi, the purple petunias, and how they were able to basically knock out the petunia pigment production by providing an RNA molecule called RNAi? That is antisense technology. They've developed a drug for viral eye infections, fomivaricin, which is antisense RNA. I mentioned bedaquiline earlier. This was improved in 2013 to treat multi-drug resistance TB. And that's a problem because about 30% of the population has latent TB. And this targets a bacterial ATP synthase. So a little bit novel strategy there. Down the road, what are they looking at? Well, there are some proposals for developing nanotube technologies where they can actually use nanotubes to poke holes in bacterial cell membranes, using monoclonal antibodies to deliver photosensitive chemicals to bacterial cells, and then when those photosensitive chemicals are exposed to a particular wavelength of light, It activates them to produce reactive oxygen species that will kill the bacterial cells. And there's also been some research done recently on trying to develop drugs which will jam quorum sensing signals in a biofilm. Remember, in biofilms, the bacteria that are in the biofilm start sending all kinds of signals to one another, and those signals can really increase the production of byproducts which result in those organisms becoming more pathogenic. So there's some interesting ideas out there in the future about new strategies that might be employed. Do antibiotics work on viral infections? Not antibiotics, and particularly not the protein synthesis inhibiting antibiotics or the cell wall synthesis inhibiting antibiotics. So penicillins, tetracyclines, etc., no effect. Okay. Are there drugs, however, which are antiviral drugs? And the answer is absolutely. Here are some examples of various types of virus that we're developing antiviral medications for. The name of the agent, the mechanism of action, and the result, basically how it interferes with the virus replication. Some of the targets are kind of obvious. Drugs to try to treat influenza virus are very important. Drugs to treat HIV are quite important. There are a number of herpes virus drugs and shingles drugs, which are quite important as well. Some of these you may have heard of before, others you may have not. Tamiflu, for example, is probably a drug you have heard of before.
Does the suffix of VIR disease viruses?
Yes. Mm-hmm. Yep. So there should be, for the most part, there are VIRs somewhere in this. Okay. Let's take a look at HIV for a second, because HIV has been in the news for a long time. There are several different strategies that can be used to attack HIV. In the HIV life cycle, HIV is an RNA virus. And during the life cycle of infection, one of the things that has to happen for the virus to replicate is that RNA has to be converted into DNA. Remember what that particular enzyme is? Reverse transcriptase. Reverse transcriptase is a convenient target to try to selectively exploit, because our cells don't have the reverse transcriptase enzyme. So there are a number of antiviral drugs which are reverse transcriptase inhibitors, which are good targets for treating viral infections. In the HIV life cycle, when RNA is converted into DNA, the DNA is then integrated into the host chromosome. It forms a provirus, and it can lie latent in the chromosome for years. That's why there's oftentimes a lag between infection and developing symptoms of AIDS. There are enzymes called integrases, which allow the DNA to become inserted into the host chromosome. So there are drugs that interfere with this integration, anti-integrase drugs. At some point in time, typically what happens is something kicks on replication of this dormant DNA, and then the DNA is transcribed into more RNA to produce more RNA virus. Well, there are some drugs that have been around for quite a while, including a drug called Acelivor, which are called nucleoside analogs. They look like a nucleotide, but they lack a 3' hydroxyl group. I don't know if you remember, but as DNA is being replicated, the new nucleotide that's being incorporated into the existing chain has to have a 3' hydroxyl group to attach to. So if you incorporate a nucleoside analog into your DNA, that basically causes chain termination. And the DNA strand stops being replicated. So these are pretty powerful drugs. Do these particular drugs interfere with DNA replication in the host? And yes, to some degree they do. But the enzymes, the viral enzymes, are a lot less discriminating than your polymerases are. Your polymerases don't have a tendency to incorporate these nucleoside analogs. But the viral polymerases are much more willing to do that. They're much less specific. And so they're really pretty good drugs. Typically, as the virus produces proteins from its RNA to make the capsid, for example, many times those proteins have to be cleaved before they're functional. And so there are a number of viral proteases which are required for the viral replication life cycle to occur. So this represents another opportunity for inhibition of the life cycle if we can develop drugs that are protease inhibitors. And again, because this is part of the viral life cycle, less likelihood that these drugs will have an effect on the host cell that the virus is infecting. What's the first thing that has to happen in order for a virus to infect a cell? What's the very first thing that has to happen? It's got to attach to the cell and get into it. Okay? So there are also some drugs that tend to inhibit entry of the virus into the cell, or uncoating the virus once it's in the cell. Okay, that's a very important step in the virus life cycle. Is once the virus gets into the cell, you've got to remove that capsid to expose the nucleic acid of the virus to the cytoplasm. That's called encoding. There are some drugs that block receptors on the surface of the virus. There are some receptors called GP120 and GP140 that look for particular receptors on the surface of a cell. And if those receptors are blocked, the virus can't attach. I was reading something the other day, and it says if you think about virus infection as being analogous to opening a door and moving through it, what these receptors being blocked really does is it's like taking the doorknob off the door. Okay? You can't open the door and get across. Another thing that has to happen is the viral membrane in an envelope virus has to fuse with the membrane of the host cell. And there are some fusion inhibitors. So the virus attaches initially by its receptors, but the membranes don't fuse. And they made the analogy that this is like, okay, the doorknob's there, you can grab onto it, but the door is stuck. Okay, you can't get through that stuck door. So because of the unique life cycle of the HIV virus, there are numerous places where you can potentially selectively inhibit the virus life cycle and hopefully leave the human unharmed. Now, unfortunately, viruses are developing resistance to many of these drugs. To try to slow down the process of resistance, typically not just one drug is being used at a time. HIV treatment usually results in or involves a triple cocktail where 3 drugs are prescribed simultaneously. The most common triple cocktail are 2 reverse transcriptase inhibitors, and then that's combined with either a protease inhibitor or an integrase inhibitor. But there's all kinds of different combinations of triple cocktails. What we're really finding out about HIV is that it isn't the death sentence that it once was, in part because of the development of all of these antiviral drugs. We're looking at HIV treatment now as being more of a chronic disease management as opposed to a death sentence. And we're not really talking about cures as much anymore as we're talking about chronic disease management. Unfortunately, one of the things that gets in the way of disease management using drugs is the cost of the drugs and their availability, particularly in parts of the world where HIV is most rampant. And that's where big drug companies have fallen under a lot of criticism is in Central Sub-Saharan Africa, for example, they just can't afford the basic HIV drugs that are needed to effectively manage this particular disease. Influenza is another disease where treatment can be really important if an outbreak develops. Some of the worst pandemics in the world, including the 1919 Spanish flu virus, have been devastating and killed millions. And so development of antiviral drugs like Tamiflu are very important. The problem is that there's a very small therapeutic window when you can provide these particular drugs and they will still have an effect. Because if you let the viral infection go too long unchecked, it's going to be impossible to wipe out. And then it's just going to have to take its course. So usually you've only got about maybe 3 days between diagnosis and when you can apply the drug and it's going to be effective. And unfortunately, things like influenza oftentimes manifest themselves as, well, it's just a cold or some kind of other respiratory infection, a bronchitis or something. And so during flu season, you really do have to be careful, and if you develop flu-like symptoms, you really need to get them checked out because those antiviral drugs can be very useful if applied when necessary. And yes, you should also get your flu shot. You really should. Okay, if you would do me a favor for a second, let's wrap up here. If you would, in your area, kind of move everything to the kind of the middle of the table and kind of organize it a little bit so we have the working part of the table pretty free. If there's anything you can put away, Bunsen burners or any other detritus that's out there in your drawers, that would be useful. And then make sure you get things out of here by 1:30.